Electron microscopes are expensive to build and maintain, but the capital and running cost of confocal light microscope systems now overlaps with those of basic electron microscopes. Microscopes designed to achieve high resolutions must be housed in stable buildings, with special services such as magnetic field cancelling systems. The samples largely have to be weighed in vacuum, as the molecules that make up air will scatter the electrons. An exception is liquid phase electron microscopy, using either a closed liquid cell or an environmental chamber, for example, in the environmental scanning electron microscope, which allows hydrated samples to be weighed in a low-pressure, wet environment. Various techniques for in-situ electron microscopy of gaseous samples have been developed as well. Scanning electron microscopes operating in conventional high vacuum mode usually image conductive specimens. Therefore, non-conductive materials require conductive coating. The low-voltage mode of modern microscopes makes possible the observation of non-conductive specimens without coating. Non-conductive materials can be imaged also by a variable pressure scanning electron microscope. Small stable specimens such as carbon nanotubes, diatom crystals, and small mineral crystals require no special treatment before being examined in the electron microscope. Samples of hydrated materials, including almost all biological specimens, have to be prepared in various ways to stabilize them, reduce their thickness, and increase their electron optical contrast. This process may result in artifacts, but these can usually be identified by comparing the results obtained by using radically different specimen preparation methods. Since the 1980s, analysis of cryofixed vertified specimens has also become increasingly used by scientists.